Hello, I'm Jake. Today, Jagruti, Varsha, and I are going to be talking about cell signaling, and specifically a paper we found that's titled Circadian Pattern of Melatonin MT1 and MT2 Receptor Localization in the Rat Suprachiasmatic Nucleus. So why did we choose this paper? This paper discusses how the location and distribution of the receptors tied to melatonin can vary throughout the day, correlating with the circadian rhythm. Not only do we find this interesting and relevant to cell signaling, but we thought it'd be interesting to actually study how the cell signaling process could influence the receiving cell, not just the cell that's producing the signal. So real quick about the authors, Nurmian E. Whaley is a PhD MD and works in the Department of Physiology within the School of Medicine at Asyut University in Egypt. And Richard Hallworth is a PhD and worked in the Department of Biomedical Sciences at Creighton University.
Cell signaling is the process in which cells communicate with each other within the body or with the external environment. In endocrine, a cell targets a distant cell through the bloodstream, an example of which would be hormones. In paracrine signaling, cells secrete local mediators that affect immediate surrounding cells. However, in neuronal cell signaling, neurons communicate with each other to transmit information. Targeting cell signaling has proven extremely successful in cancer treatment. Drugs such as Gleevec, Herceptin, and Idasa are being used to treat millions of patients worldwide. This development is mainly because of the understanding of cell signaling pathways, cancer cell metabolism, studying receptors, and their impact on cancer cells.
The introduction section of this paper starts with some background information on melatonin and its regulation. Melatonin is a neuroendocrine hormone secreted by the pineal gland. Its synthesis and secretion is regulated by circadian rhythm, and circadian rhythm is in turn regulated by the suprachiasmatic nucleus. So as we can see from the figure on the right, the light-dark cycle, which is pretty much the cycle that allows us to know whether it's day or night, will provide visual input to the suprachiasmatic nucleus, which paired with the pineal gland helps regulate the amount of melatonin secreted into the body. These levels of melatonin in turn provide feedback through modulation of electrical activity of the suprachiasmatic nucleus via these MT1 and MT2 receptors. What are these MT1 and MT2 receptors? They're the melatonin GPCRs, and there's not a lot of information known about them. Specifically MT2, where it's more difficult to evaluate its mRNA, and there's low levels in this suprachiasmatic nucleus. The reason there's not a lot known about these is a lack of specific antibodies that are used for them today. So this study will then look at the circadian distribution of the MT1 and MT2 receptors in the suprachiasmatic nucleus. This should help shed light on the temporal gating of melatonin efficacy in modulating behavioral rhythm. Here's the methodology followed within the study. They started with animal and tissue preparation, followed by immunohistochemistry, then immunoblotting, immunofluorescence, and they finished with image analysis. The way the study was set up is they had rats 4 to 6 weeks old that were trained for at least 2 weeks with 12 hours of light, followed by 12 hours of darkness, in continuous cycle, and then at dusk, 4 different rat brains were sampled. I'm going to skip through some of the finer details that were involved in the next few steps, but it pretty much involved blocking nonspecific binding sites for melatonin, and then finding the specific proteins involved in the receptors in MT1 and MT2, and then in turn the specific antibodies that were used to target and image those specific receptors. Majority of these steps just involved more information on how the samples were stained and washed and prepped, and a lot of things that were outside of the scope of this class. And then they finished up with some image analysis, and they used statistical analysis to find significant differences between the areas that they were looking at and the densities of the receptors in those areas based off of pixel density.
Coming to challenges that were faced, a circadian rhythm of receptor protein synthesis and localization could not be demonstrated due to the lack of specific antibodies for MT1, MT2 receptors. So to overcome this, more research can be done to find out the specific antibodies as their rhythm would explain temporal gating of melatonin efficacy in modulating behavioral rhythm and electrophysiology. Let me give you an insight to the results obtained. The images here show MT1 receptors localization in the cortex and hippocampus. In the hippocampus, labeling was found in CA1, CA2, and CA3 regions, mainly around the cell bodies. In the neurons, the labeling appeared to be restricted to the cell bodies and not the processes. The images A and C show low and high magnification pictures of label of hippocampal neurons with anti-MT1 antibody. B and D show low and high magnification image in cortical neurons. There is no circadian change in pattern of MT1 receptor expression. Here we can see the pattern of labeling for MT1 receptors within SCN, which is examined across the circadian cycle. MT1 receptors were detected within SCN at dusk. Dense labeling and a diffuse pattern at circadian time was observed. Moreover, the labeling also extended up to the optic chiasm, which also meant the probability of labeling neuronal dendrites. There was no difference in distribution of labeling within each SCN. Labeling was observed during other three circadian times, but was less pronounced as compared to dusk. Also, the pattern of labeling was more discrete, probably labeling only the cell bodies of SCN neurons. Using one-way ANOVA quantification method, it was observed that there was a significant variation in MT1 receptor density in SCN across the circadian cycle compared to non-SCN areas. Average MT1 receptor density in SCN was increased at dusk as compared to dawn and midnight. The average density of MT1 receptor label was higher at dusk than at midday. On the other hand, MT1 receptor density compared to dawn and both midday and midnight was statistically insignificant, although the label was greater at dawn compared to other times.
From the results observed, the cyclic pattern of melatonin receptors expression and degradation proves that melatonin was most effective in influencing SCN function only at specific windows of time. This shows the possible feedback of melatonin on the SCN. Melatonin is secreted in darkness by the pineal gland. Light inhibits melatonin secretion by decreasing the activating influences of neurons in the paraventricular nucleus. The neural pathway from the paraventricular nucleus to the pineal gland is circuitous, passing through the spinal cord and superior cervical ganglion. Melatonin binds to the receptors on the SCN and decreases the alerting signal during darkness, thus promoting sleep. This paper shows the first demonstration of the daily pattern of melatonin receptor protein expression using the immunohistochemical approach. Some of the study improvements are the sample size should be increased as there is no apparent daily change in MT1 labeling in the cortex and hippocampus. Moreover, MT2 receptors were not detected in the study. There is the possibility that the level of MT2 receptors in the SCN is not sufficient to be detected by this method at any time. Hence, advanced quantitative methods are required to confirm these observations. Conclusion. Understanding the role of melatonin is important for the regulation of circadian rhythm, as it helps in using this hormone in jet lag and sleep disorders. The use of melatonin in jet lag appears to decrease jet lag symptoms. Moreover, melatonin may also be helpful in rotating shift schedules to improve sleep quality and maintain normal circadian rhythm. In some patients with insomnia, melatonin appears to induce sleep onset. A recent study also suggests that melatonin would be the most effective agent for preventing or treating COVID-19. Further investigations and studies in understanding the role of melatonin is important for the regulation of circadian rhythm as well as eradication of diseases such as COVID-19, sleep disorders, cancer, and many more.